All right, well, we'll get started. Hi, everybody. Welcome to this webinar. So I just wanted to introduce our speaker, Dr. Florian Castet. Dr. Castet graduated St. Peter's back in 2008, received several prestigious awards while in medical school, and specialized in medical oncology at the Institut Català d'Oncologia by pursuing a master's in clinical oncology and molecular oncology. Now, for those of you who are not sure what oncology is, it is the science of cancer. Dr. Castet is currently working at the Liver Translational Research Group and is exploring the molecular and immune implications of liver cancer. Now, most of us know that the immune system's role is to identify different pathogens and try and destroy them to protect us. But the notion that it can also have the potential to detect cancer cells is something that we don't really learn about, at least not in high school science. And I remember I first heard about it when a previous US president, Jimmy Carter, who was well into his 90s and had advanced brain cancer, was cured by immunotherapy. And this is definitely something that is interesting and hopefully a future treatment or even current treatment for cancer patients all around the world. So without further ado, I will let Dr. Castet guide us into this promising scientific path.
Okay, thank you very much for the presentation. And I'd also like to thank Joan Rovira for inviting me to take the presentation, and for Patricia Maragos and Nuria Garcia for their help in the preparation of the presentation. So today, as Patricia said, we're going to talk about how we can harness the immune system to treat cancer. Now, many of you are probably aware that there are many different treatments for cancer, which includes surgery, radiotherapy, and chemotherapy. But lately, immunotherapy has been introduced as a new tool for trying to treat cancer. And we'll talk about why it has become so important and what is its great potential in this area. So I have divided the presentation into these three big blocks. First of all is briefly telling what cancer is. Many of you are probably aware of it, but I'd like to emphasize why it is so difficult to treat cancer and why many cancers are considered incurable still today. Then we'll talk about the immune system and specifically focus on those areas that are most important to understand how we can then use it to specifically target cancer. So at the end of each of these sections, we'll leave a time for questions. So feel free to ask any question that you are interested in, and I'll try to answer. So the first, very briefly, you know that cancer comes from the Greek word meaning crab. And the reason for which it's called that way, it's because when you look at how cancer looks on a radiography, which is here shown, this white part here is what cancer is. And you can see that it has some prolongations here, which look like crab's legs. And these prolongations, what they reflect is cancer's aggressive behavior of how it goes into normal tissue and destroys all the surrounding normal cells in here, for example, in breast. Oh, sorry. So in the year 2000, there was the first breakthrough in understanding how cancer works was by introducing what is called the hallmarks of cancer. And these are traits or characteristics that make cancer cells aggressive and that make cancer cells proliferate. But basically what you could sum up these 6 characteristics is, first, an uncontrolled cell growth. That means that cells divide and proliferate without any type of control from normal stimuli in the body. And then they acquire a very aggressive behavior. They are able to destroy the surrounding tissue. And more importantly, they're able to go into other organs in the body, such as liver, brain, or lungs, what we call metastases. And that is very important because even today, 90% of deaths in cancer, that is 9 out of 10 deaths in cancer, are due to metastases. So these same cells are the ones that come from normal cells and eventually acquire these hallmarks, we would say, to become this aggressive and proliferating cells that will eventually kill the patient. So this is what I want to focus on. That cancer evolution gives rise to tumor heterogeneity. And there are many long words here, but we're going to explain it now. But it's important to understand this because it's one of the main reasons why cancer is so difficult to treat and why cancers, once they spread to other organs or give way to metastases, is considered in most cases uncurable still today. So now imagine you have 3 normal cells in whatever organ in the body. And these are normal cells, meaning that they proliferate under the control of the normal stimuli in the body, whether it is cytokines, whether it is hormones, whatever it is, they're proliferating in a controlled fashion. And by organizing correctly, they are able to make the organ function. Now, imagine that due to whatever risk factor, the most common one is tobacco, one of these cells acquires a mutation in a gene that is very important and that gives this cell a different behavior or phenotype to their surrounding cells. This cell will be able to proliferate more, and this cell will have enhanced capacities when compared to the other cells. So if you apply here Darwinian evolution, what you know is that eventually these cells will proliferate more than their normal counterparts. But even so, because they proliferate more, they have a greater risk of acquiring new mutations, not by any, whether it is by tobacco, but also simply because it is proliferating more. They have a greater risk of acquiring a new mutation. Now, suppose that a patient is diagnosed at this stage here. So you take a biopsy and you see these red cells. So what the oncologist decides is that we're going to give a specific treatment that targets the cells which look like the most predominant cells. So they give a treatment, whether it is chemotherapy or other types of therapies, to target these cells. Now, what will happen is that these cells, because they are being specifically targeted, will decrease. But because we are not treating the blue cells, these will increase. And because what we said before, they will proliferate more, and also a new type of cell will appear with a new mutation. So now the oncologist will decide to change treatments to treat the blue cells, but this will give rise to even more phenotypic heterogeneity. So eventually what we are doing is by giving evolutionary pressures to the cancer, we are giving rise to a very heterogeneous tumor with many different cells that will spread to other organs. And when this stage appears, because there is so many different cells to treat, there is no treatment that can target them all. And that's why many cancers today are incurable once they have metastases. So if we follow on, in these same authors that talked about the 6 hallmarks of cancer in year 2000 and 2011, they proposed a new set of hallmarks of cancer, one of which was avoiding the immune system. And so that's what we're going to focus on today. So before going into that, I just want to review some specific aspects of the immune system that is interesting for cancer. But before, does anyone have any questions until now of what we've said, or something that has not been clear? Okay, I'll go on for the moment, and if any question arises, we can give them at the end. So about the immune system, what is important to understand is the theory of immune surveillance. Now, this theory was proposed early in the 20th decade, in the 20th century, by Paul Ehrlich. And what he proposed was that the body constantly generates cancer cells that do not reach to become a tumor because they are eliminated or detected by the immune system. So what he was saying actually was that in most human beings, some cancer cells will appear during their lifetime. And this has been proved to be true. Almost all human beings have some kind of cancer cell in the body at least one time in their lifetime. So how does the immune surveillance system work? Well, with the knowledge we have today, most of it depends on the adaptive immune system, which, as you know, is the one that depends on T cells, B cells, and the antibody producing plasma cells. So the immune surveillance system has three phases, three stages that are very important to understand. First, there is the stage of elimination. That is when the premalignant, meaning cells that will become cancer eventually, but have still not acquired this aggressive behavior, these cells are initially recognized by the immune system and eliminated, rendering this abnormal tissue normal again, a normal epithelium. But eventually, it will give step to the second stage, which is the stage of equilibrium or the stage balancing, meaning that the immune system is strong enough to be able to recognize the premalignant cells, to eliminate most of them, but some of them remain. And so the immune system and the premalignant tissue are together, we could say like living together in this balance. But then in more advanced stages, these cells will acquire what we call mechanisms of immune escape, meaning that these cells will be able to proliferate and escape the surveillance of the immune system and will eventually give rise to metastasis, or this very aggressive behavior that we were talking about before. So why would we want to use the immune system to treat cancer? Now, there are many characteristics which make it interesting, but there are three which I want to highlight. First is that the immune system is highly specific. It is able to differentiate a cancer cells that comes from normal cells from its normal counterpart. If there is one single protein that is different, the immune system is able to recognize this difference, and therefore it could target the cancer cell and not harm or hinder the normal cells, which is what we're not able to do with chemotherapy. And that's why so many secondary effects or adverse events appear with chemotherapy. Then the immune system is durable. It has a good memory, and therefore it could potentially prevent any long-term persistence of the cancer or any long-term what we call recurrence. That means once you treat the cancer, it will not come back. And then, and most importantly, adaptable. That is for what we were saying before. As the immune system is able to adapt to changes in the cancer, it could potentially break this cycle of tumor heterogeneity and cancer evolution and target all cells at the same time. That's why it makes it such an interesting player in this area of treating cancer. So now we'll go into the different types of treatments we're using by using the immune system. Any questions until now? Okay, so now we'll talk about the last part, which is how we can use the immune system to treat cancer. Now, in 2013, Science, which is one of the most famous science journals in the world and Nature highlighted the potential uses of cancer immunotherapy as something that could be potentially very beneficial for patients. But it is not something new that has appeared now. Cancer immunotherapy has been used for decades, and even from the past century. In the 19th century was the first use of immunotherapy to treat cancer. And so it was the first, the first attempts were quite, we'd say, aggressive nowadays. And it was based on the fact that some people observed that patients who had tumors in the skin and at the same time had an infection of the skin due to a bacteria such as Streptococcus pyogenes, they had spontaneous regression, meaning that spontaneously, without giving any type of treatment, the tumor disappeared. So one surgeon in New York called William Coley decided to try to use this as a way of treating patients with cancer. And what he did was take some parts of this bacteria here, and he created a toxin. He took parts of the bacteria and he directly injected that into the tumor, into superficial tumors. So he was actually inducing an infection of this cancer. And what he saw was that a certain amount of patients were cured. Now, despite this was well, the results were quite outstanding for its time. It was not implemented, probably because many patients also died as a result of infections caused by the injection of this bacteria into the cancer. But nowadays, we can use more sophisticated technologies to try to apply this in a more secure manner to be able to treat cancer, but not to generate so many problems or adverse events. And the first and most important treatment that are being applied nowadays are checkpoint inhibitors. Now, these have really supposed a breakthrough in cancer management, and this is applied every day in many hospitals across Spain to treat cancer patients. So to understand how this works, I would first like to do an analogy. If you think about Formula One race, there are areas in the race where the driver has to step on the gas pedal to go as fast as he can. That's when there is a straight line, for example. But in other parts of the race, he will have to step on the brakes, because otherwise he will not be able to take this curve correctly. So winning the race implies knowing when and how to step on the gas pedal and step on the brakes. And the immune system functions in a similar manner. In order for it to be able to work correctly, it has to be tightly regulated, meaning there are times when the immune system has to be very active, such as when there is an infection or whether it is bacterial or viral infection, and other times when the immune system must be inhibited, or what we call must stop, such as, for example, in when it recognizes normal cells in the body. When this system does not work, some diseases may appear as a result of an overactivation of the immune system, such as autoimmune diseases, lupus, or other kinds of autoimmune diseases, which I have probably heard of. So to do that, what the immune system has are co-inhibitory and co-stimulatory molecules. These molecules are expressed on the T cells, which are the immune cells that are in charge of recognizing the tumor, but are also expressed on usually on antigen presenting cells, which are those cells that digest the virus or the bacteria and present it to the T cell. So when the immune system has to be active, what the antigen presenting cell will overexpress are co-stimulatory molecules. When the immune system must not be active, the antigen presenting cell or other cells of interest may present co-inhibitory molecules. Now, why is this important for cancer? Well, because cancer uses this system to hide from the immune system. And he does it in the following manner. Imagine if a cancer decides to overexpress, that means express in very high quantities, co-inhibitory molecules. What it'll do, it will completely paralyze the immune system. And that will give rise to what we call exhausted lymphocytes. That means that these lymphocytes are no longer able to detect the cancer and are no longer able to carry out their usual function. So for you to be able to see this, I want to show this diagram here. Now, this is what you would see if you look at a cancer in a microscope. This is the tissue you would find, and depending on how you treat the tissue, you would find it blue like this. That would be what you would usually find in normal cells. Now, suppose we use PD-L1 and we use a specific antibody that will stain it in brown. Okay, here you can see that this cancer here, for example, has a very high expression of PD-L1 when compared with this other cancer here. And the reason for this is because some cancers, not all, but some cancers overexpress PD-L1, which is one kind of co-inhibitory molecules, but there are many others, to inhibit or to stop the immune system from carrying out its function, and therefore inducing exhausted lymphocytes. So this is what you would see here. The tumor cell expresses PD-L1, which is the molecule we say, and by interacting with its receptor on the T cell, is able to stop the immune system from doing its function. So some very brilliant authors decided to, what if this is one of the mechanisms cancer uses, what if we tried to block this interaction here? What you would do is you would release the brakes that the tumor cell is doing. If you're able to break this interaction here, you are no longer submitting the T cell to this inhibitory stimuli, and therefore it is able to be active again. And so what they did was design some specific antibodies that are able to recognize either of these receptors and therefore block the interaction between these two receptors. And therefore, they are able to activate again these T cells. And so this was developed in the years started in 2000, but in the clinical setting in the year 2010. And as a result of this, you can see that here, this is the example of an example of melanoma. Okay, this is what we call in medicine survival curves, which plots on the x-axis time, on the y-axis percentage of surviving patients. So basically, the higher the curve, the better the survival. So you can see here that CTLA-4, which is one kind of these antibodies that I talked about, and PD-1, which is another kind, have really rendered outstanding results when compared to chemotherapy, which is the blue one here. So just to give you a number so that you are familiar with this and understand how important this improvement has been, before these treatments, melanoma patients who had metastases survived less than a year, usually. And no patient could be cured, or it was really exceptional. Now, with these treatments, more than half patients survive more than 5 years. So we're talking about a very impressive increase in survival. And what is most important and what we're seeing now is that some patients do not have cancer after 5 years, meaning that maybe some of these patients are cured with these treatments, something that was completely unthinkable before with chemotherapy. So because this has been a real breakthrough in the management of cancer, the authors who designed these treatments and discovered these molecules were given the Nobel Prize in medicine in 2018, which was Busch and Fehleisen in the States and Tetsuko Honjo in Japan. Now, this was the first treatment, how you can use cancer, and this is used. Then there is what we call adoptive cell therapies, which has an incredible potential, but because of different reasons, is still not being used very much in the clinical setting. We'll see before because it's quite complicated. Now, remember that before we said that because cancers express these co-inhibitory molecules and because of other things, the T cells do not work appropriately. And what we called this were exhausted T cells. Now, even sometimes when this state is so profound, so important, even if you give checkpoint inhibitors, you're not able to revive these T cells. So those cancers will not respond to checkpoint inhibitors because these T cells have entered such a state of exhaustion that they are not able to be revived. So what some authors said was, what if we extracted these T cells, which we know are recognizing the cancer, and we stimulated them to give them back to the patient? So basically what they proposed was, suppose we have a patient with a cancer, by surgery, you take the cancer out, and then you sort this cancer and you only take out the T lymphocytes, what we call tumor infiltrating lymphocytes. So here you have selected these tumor infiltrating lymphocytes, which are not working because the cancer has really driven this state of exhaustion, but you cultivate these cells in the lab, you submit them to different kinds of activation by using cytokines, there are different methods to do this. Then you expand them. And after you have retrieved and reactivated these T cells, you give them back to the patient as if you were doing a blood transfusion. This has been used in some cancer types, and really impressive results have been obtained, especially in breast cancer. This has been explored, and interesting results have been obtained. But there is even a more sophisticated technique that people are using, which are what we call CAR T cells. CAR T cells are engineered T cells. So what you do in this case is you take T cells or lymphocytes from the blood of the patient. You no longer have to operate the patient. You don't have to take the cancer. You simply take lymphocytes from the blood. You do not know whether these cells are recognizing the tumor or not. But what you do is that you insert a gene that codifies for the T cell receptor. That is those cells, that is those molecules that are recognizing the cancer. And moreover, you know that this T cell molecule is an enhanced T cell molecule. So it is an enhanced receptor that will work even better than the one that is present in the body. So you transfect this by using a lentivirus. You transfect it into the T cell. Again, you cultivate it in the lab, you expand them, and you infuse them back into the patient. Okay, so they are actually, what you're doing is engineering a specific T cell from the patient and giving it back, knowing that this T cell, what it will do, will specifically recognize this cancer cell. And so here I'm presenting some incredible results with these therapies. And in this part, they are blood cancers. Okay, they are what we call lymphomas, which are a kind of blood cancer and of lymph node cancer. And in this trial, what they saw was that they selected patients that had progressed to all other lines of treatment, meaning that they had not responded to many different types of chemotherapy. And these patients that in other circumstances would have probably died in less than 6 months with no other possibility for treatment were infused these specialized engineered CAR T cells. What they saw was that the number of patients that responded, meaning the number of patients who presented a decrease in the size of the tumor, was more than half in both cases. But what is really impressive was that after 5 years of receiving that treatment, 31% and 43% in 2 different types of cancers were still alive, and the tumor had not grown. So again, the question asks whether we are actually curing those patients or not. It is probably still early to say this, but the results that these treatments have provided are really impressive, and these have been approved by different drug agencies and are being used in the clinical setting in different countries worldwide. And so the final point, which I want to talk about, is one that is quite known by many people now with COVID-19 vaccines, are cancer vaccines. And so they're very similar to what we would use for COVID-19 vaccines. What it is based on is using a vaccine that is specifically made up of the cancer that patient has. So what the authors do for this is that they take the cancer cells and they try to understand what are the proteins that are most important in driving the immune system. And they use these proteins as the vaccine. They make up the vaccine with these proteins and they then vaccinate the patient. Now realize here that we're not talking about preventive vaccines. We're talking about therapeutic vaccines, meaning that they are vaccines that are used when the cancer has developed, and therefore they're vaccines aimed at treating the cancer, not at preventing it. So the experience so far with this type of treatment is more limited than with the other two, but 2 articles published in Nature, and there are other kinds of vaccines that have been developed and published too, have seen that more than half of the patients that are treated with these vaccines, even though the number of patients is relatively small, do respond and do present a decrease in the tumor size. And even patients that do not respond when they are then treated with checkpoint inhibitors, these patients respond exceptionally well to checkpoint inhibitors, meaning that probably these vaccines are contributing to activate the immune system. But as I said, this is still being explored in the clinical setting. So as conclusions, and then we'll have the questions and answers. Cancer is a highly complex biological process, and still today it remains a difficult to treat disease. But immunotherapy has triggered a paradigm shift in cancer treatment and has prolonged survival in some patients. Not in all. We are not able still to select appropriately which patients will respond or not, but in some patients, they have responded in an exceptional manner. Different strategies are being used, which we have described: adoptive T-cell therapies, checkpoint inhibitors, or vaccines, and there are many other strategies which are being pursued, but we could not look at them all. It would have been good, and new strategies that combine different approaches, like combining vaccines with checkpoint inhibitors, are likely to impact patient survival and are likely to contribute. And maybe one day we may talk about cancer being cured, even when metastases have developed. So now I will be glad to take any questions if you have any.
For the Q&A, I think it would be nice for Florian to be able to hear your voice. So if you have a question, please feel free and unmute your microphone and even show your face in the camera. That'd be great. And ask your question. And if you don't feel too comfortable with that, you can always write the question in the chat.
What are the signs that somebody has cancer?
It's quite complicated to say because every cancer is different. It is very different if you have a cancer in the lung, where probably some of what patients will have is dyspnea or breathlessness, difficulty to breathe, or pain, than if you have a cancer in the brain, for example, or in the liver. So globally, almost all cancers usually have fatigue, meaning that they're usually very tired. They lose weight, and usually patients lose their appetite. They do not eat that much. So these would be like 3 of the cardinal signs, but then there are many other that depend on where the cancer is located and if there is metastases or not. Have I answered your question, Sophia?
Yeah, thanks.
Okay, thank you.
If immunotherapy works against a certain type of tumor, could we eventually develop a vaccine for them and prevent the development of tumor even before it develops?
Yes, that's a very good question. The problem with preventive vaccines is that you do not know who will develop cancer. So giving a vaccine to everybody would not make much sense, because it's not like COVID-19 where everybody will eventually be exposed to it. We do not know who will develop cancer, but even more importantly, the vaccines we are talking about now and we have now are specific vaccines that only work for one kind of cancer. And we're not saying for lung cancer, we're saying for lung cancer in a certain patient, because we are doing this vaccine based on proteins that are expressed in that cancer. So we would not be able, with the current treatment strategy, to develop any preventive vaccine, because we would not be able to know what protein this cancer will have until we're able to see it. If science advances, and let's hope it does, maybe one day, but with the current treatment strategies that are being implemented, nowadays it is very complicated.
I have a question about the vaccines. I believe that vaccines are very expensive to make, right? So if cancer vaccines had to be personalized for every patient, I was wondering how much work the whole aspect of vaccines, how much work needs to be done in there so that cancer vaccines become available to pretty much everybody.
Yes, that's a very important aspect to look at, and very good question. Almost all new treatments are very expensive, and that is one of the great limitations and why they're not being implemented into the clinical setting as fast as we would like. Not only vaccines, but if you think about checkpoint inhibitors, which these are not personalized, you give it to most patients, each injection costs about 3,000 to 5,000 euros. And these injections are given every 2 to 3 weeks. So if you do the calculations, after a year, you're talking about several tens of thousands of euros for only one patient. If you go into personalized therapies like adoptive T cells therapies and the CAR T cells that I talked about, this is even more expensive. Because this, as you say, is personalized, is one treatment per patient, this costs about only the treatment, 500,000 euros, only the treatment. So then if you think that for doing this treatment, the patient has to be hospitalized because there will be problems, because almost all patients develop problems with these kinds of treatments. The total amount may reach sometimes 1 million euros for a single treatment. So this is a problem. And this is one of the reasons for which it is being very difficult for agencies to approve these treatments, because it is the government and patients cannot pay for it. It is so expensive. And it always happens that when science develops something new, the initial costs are extremely high. Usually they go down with time when the technology is improved, when it is easier to implement, when more laboratories, and when what they call the patent disappears, the costs usually go down. But nowadays, it is something quite unaffordable. So there is, I do not have a solution for that. It is very difficult to say.
I have one question.
Mm-hmm.
Does like the new strategy work better than all the other strategies before?
Pardon, I didn't hear the last part.
Does the new strategy of curing cancer work better than all the other strategies that were?
No. So immunotherapy is just as another tool that we are using to treat cancer, but it is not substituting the other tools we have. So for example, still today, when a tumor is small, the ideal treatment is surgery in most cases. And there is nothing that matches surgery in those cases, because you can cure small cancers by simply operating them. What we are,
okay, thank
you. What it is being useful, it is to combine it. For example, if we give immunotherapy before surgery, we know that many cancers go better than if we gave only surgery. And also we can combine, and this is being used, combining chemotherapy with immunotherapy. We know that this, for example, in lung cancer, is better than giving only chemotherapy, and in some cases that giving only immunotherapy. So this talk was more about introducing the concepts, but in the clinical setting, before using a treatment as immunotherapy, we have to be sure that it is better than all other strategies. And we do this by doing clinical trials. If there is no clinical trials, we do not implement the strategies here. So the only ones that have been approved here are checkpoint inhibitors and CAR T cells in some blood cancers. In all other, we still use all the other tools that the chemotherapy, radiotherapy, etc.
I have a question.
Mm-hmm.
When engineering CAR T cells, how can you know which is the specific type of receptor that is detecting the cancer? I mean, is it always the same, or are there different receptors?
No, it's not always the same. That's the very interesting part of doing that. What you do is you introduce a gene of an artificial receptor that activates far better than the ones that are usually present surrounding the cancer. So we are able to introduce this gene into the patient's lymphocyte. And then by introducing this gene, it expresses and it's able to recognize the cancer by this way. So what we are doing is putting a receptor that recognizes what we call neoantigens, which are proteins expressed in the tumor that give way to the activation of these T cells. And the good thing of this receptor is that it is highly more probable for it to activate the T cell when compared to a usual receptor that is exposed to the cancer or the lymphocytes that are present in the cancer.
Okay, thank you.
Welcome.
There was a question in the chat, and the question is, it segues a little bit to what Isabel mentioned about accessibility and cost. The question is from Jenny, and she writes, is it an accessible technology? Could it be applied in developing countries? And then could an ethical debate arise from it due to the technology involved to apply it?
Yes, that is very complicated to answer. Now, most of these treatments are not being used in developing countries. Basically because in many of these countries, there is not even access to basic antibiotics that cause many more deaths than cancer. Now, many children in these countries are dying because of pneumonia or tuberculosis, where we have very effective and curative treatments, but these treatments are not able to reach these countries because of different factors and lack of political will. But these treatments are very unlikely to be today to be implemented in these countries because they require a very important infrastructure, especially for adoptive T cell therapies, apart from what there is. They require good hospitals, ICU, very important treatments, ventilators, and these are not available in these countries because the risk of there being problems with these treatments is very high. So even if the cost could be afforded, which I don't think it is affordable right now, it would be very complicated because they do not have the infrastructure for that. So yes, I think it gives rise to ethical aspects, but I think that it is part of the same problem of why are curative antibiotics that are not expensive not reaching those countries where they are most needed. I don't know, that is personal opinion. That is not something factual.
Is there any side effects to the immunotherapy?
Yes, there are. There are many side effects. Each treatment has different side effects. Overall, checkpoint inhibitors are usually considered to be better than chemotherapy. There is less side effects because of what we said about being specific. But if you think about how the treatment works, we're saying that what we're doing is releasing the brakes of the immune system. So if we're releasing the brakes of the immune system, it is not only in cancer, but we are maybe releasing the brakes in other organs in the body. So what we have seen these treatments can cause is inflammation in different organs. It can cause inflammation in the lungs, in the liver, in the skin. And the severity of this can go from simply having an itchy skin to some patients even dying despite receiving all the treatments to try to counteract this. And when you talk about the adoptive T cell therapies, the patient has to be hospitalized because there is severe adverse events. Maybe you have heard of COVID-19 causing an inflammatory syndrome. That is a cause for it having to go into hospital well, these treatments cause the same thing. They cause this type of inflammatory syndrome. And when this appears, they have to go to the intensive care unit, the ICU. But the benefits outweigh the problems.
Thank you.
You're welcome.
So there was also a question from Christina, kind of along the same lines of side effects, but do the vaccines have any side effects?
Yes, now the vaccines, they're still being explored. The small trials I showed did not have significant adverse events, but the vaccines were developed in early 2000s, I think, and they had to be stopped because they had severe adverse events, because they were trying to do them with viruses, and these viruses were inducing problems in the cells. They were infecting the cells, and so the experience with these vaccines is not as important as it is with the other treatments, but it is expected to have a similar profile of adverse events when compared to checkpoint inhibitors or to adoptive T-cell therapies. Maybe not as severe, but probably we will see the same thing.
And another question from Martina. Her question is, do the CAR T cells work against every type of cancer?
Well, we know that against certain types of cancer, it works very well, such as the blood cancers. Cancer of the blood, they work very well in many of them. And in other tumors, it has not been as successful, and it has not been as, I'd say, as good as in blood tumors. But it is true also that usually blood cancer always goes a step ahead of all the other types of cancer. So probably we have to improve the technology, and it will eventually work in other types of cancer. But it is true that there are some kinds of cancers that do not work well with immunotherapy, with checkpoint inhibitors, such as testicular cancer or pancreatic cancer or other types of cancer. We know that they don't work as well with immunotherapy. So a lot of efforts are being aimed at trying to curb this resistance and make immunotherapy improve the efficacy in these cancer types.
A question from Nafeli. How do you know the cancer has left the body, the person has been cured from cancer?
Well, it is very difficult to say. Usually there is not a universal definition of cure. Usually for considering a patient to be cured in most cancers, what you say is you have to have a disappearance of all tumors by using different imaging techniques, radiographics or CT scan or PET CT scan or other techniques. And usually we say that it has to last for at least 5 years. But in other cancer types, you extend this to 10 years because there are what we call late recurrences, which means that once the cancer has disappeared, it may appear again after many years since the initial surgery or something like this. So that there is not a universal definition of cure. And there are even some cancers, it is rare, that come back after more than 10 years of surgery. So it is difficult to say, but usually you say after 5 years, if you have no other tumor in the CT scan, you can consider a patient cured.
A question from Noah. How do you determine which treatment to use in each cancer?
Wow, that is very complicated to answer. It is very long, let's say. Basically, what I said before. It is based on clinical trials. So clinical trials, what you do is compare one treatment with the other and see which one is better. So usually you do some for each cancer type. So if it goes better for a cancer, then you use that treatment. What we're seeing is for immunotherapy, for example, what I talked about PD-L1, which is the checkpoint inhibitor, co-inhibitory molecule, we are looking at the microscope if there is a lot of PD-L1. And in those cases, if there are a lot of PD-L1, we know that if we give a specific antibody, these patients respond exceptionally well. This is what we call a biomarker. When you have some kind of information that tells you what patient will respond to what kind of treatment. So in those cases, we usually give these anti-PD-L1 treatment. But this is different for every cancer type. So there is no universal rule of knowing how to treat each cancer. There are specific protocols and guidelines for each cancer type. And with immunotherapy, we're trying to find ways of predicting which patients will respond. But it is still not very, we have still not been very successful at it.
We're almost running out of time. Are there any last questions for Florian?
Pat, I just wanted to ask a question just in relation to the adoptive cell therapies. Florian, you were saying that in this stage, would this be in late stage of breast cancer, then that you'd extract the tumor and then separate the cells, and you were saying the T lymphocytes from the tumor, and then you'd cultivate them in the lab, you were saying, and then you would return them to the patient. Would that be more for preventative later on in case the cancer came back, or is it just for treatment?
Well, in both ways, probably. Nowadays, because of the secondary, the adverse events and the possible side effects you can have with these therapies, they're only being used in patients who have a dismal prognosis, or what we'd say a very aggressive cancer, and it's only being used in those circumstances, because otherwise the risks outweigh the benefits. So, but in those patients, what we have seen is that some of these cases have a complete remission of the cancer, which means all the tumors disappear. And we know that in those cases, those that still have lymphocytes in the blood of the original ones you infused are the ones that will most likely not have a recurrence or the appearance of a second tumor. So if we are able to make this procedure more standardized, to make it more secure, to make it with less side effects, this could also probably be used in the context after surgery for preventive intent. But
one more question: what time frame would you be talking about then? By the time the cells are extracted and they're reinfused then or re-injected into a patient, like how long would that take for a patient, let's say, because you were saying you need to be hospitalized for this treatment? So are you talking 6 months in hospital, or?
Well, normally you first extract the tumor, then the patient can go home. You then do the work in the lab, which takes about, it depends on which one, but for CAR T cells, for example, it takes about 1 month. And then the patient would come back, and then the patient will stay as long as is required. Usually it will be about 1 month, but sometimes it can go longer. This is similar to what patients with leukemias do when they do intensive ablative chemotherapies. These patients usually stay in the hospital for 1 month. So it would be something similar to what they're doing in hematologic malignancies.
Right. Okay. Thank you.
Welcome.
Nuria?
Thanks. I also wanted to ask you, because you were saying then, Florian, before when we were talking that, and also now you were explaining about CAR T cells and how important lab work is, but you were also mentioning that a lot of things are investigated as well with bioinformatics, and I would like to stress that, because I would like also the students to know that it's not only lab and the biology basis that it's needed, but it's also a lot of work that it's done, which involves our use of technologies. So if you could explain a little bit on that, thank you.
Yes, for example, the vaccines, when they design the vaccines, the way of being able to know what protein will be most effective is they try to predict, computationally predict, how these proteins will be recognized by the immune system. And they use many different algorithms, but basically what they try to do is compare these proteins with some viral proteins, which they know really activate the immune system. And by doing that, they're able to infer what proteins are most likely to be recognized by the lymphocytes. There are many also spin-offs, what they call spin-offs, which are small biotech companies, such as the one that developed the Pfizer vaccine, that are aiming their efforts at specifically developing this kind of technologies to be able to predict it. And in fact, one of the authors of the vaccines in cancer was one of those. It was Ugur Sahin, which is the same one that developed the Pfizer vaccines. So it is really something they're working on to, and many collaborations, many institutes, academia, and many small biopharmaceutical companies are trying to develop this, and a lot of it is based on bioinformatic analysis of large data sets of big data, because now what you're able to have by sequencing tumors is big data, is huge databases which you can only analyze with very advanced statistical methods and bioinformatics.
Just to finish off, this is a personal question, if you don't mind. So what led you to the specialization of oncology compared to other specializations that you can choose from?
Well, I really liked, there are many aspects in oncology which I find very, very interesting. It is a very challenging specialty because there's still lots of work to do and lots of aspects to develop. I find it from a biological perspective fascinating of how the cancers are able to adapt to different treatments and how they're able to overcome all the usual. And by studying cancer, we have been able to understand in a far more profound manner how the biology in the body works. And then from a medical perspective, it is really gratifying. It is very difficult in the clinical side, but when the relationship you establish with the patients, I think is quite privileged when compared to other more not as, I say, when patients go to a specialist and just see him once, you're not able to establish a bond. In oncology, because you have to follow up the patient for months and in most cases years, and you see him once, twice, or more times a month, then the bond that is established is I'd say unique to other specialties.
All right, well, on that note, I just wanted to thank our speaker, Dr. Castet. Thank you so much for enlightening us on the aspects and the promise of immunotherapy. I'd like to thank all the participants as well. Thank you. Everyone who joined and asked questions, thank you so much, and have yourselves a very good evening.